Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic crosses in Drosophila regarding 100% female lethality in Carly mutation in Drosophila. You have to remember that this 100% female lethality is also known as double lethality. Okay, double lethality cross. Now, before beginning, I would like to request you to kindly watch my 50% female lethality video regarding this so that you can understand this video very easily. Okay, I will provide the link of that video in the description box below, and I will also attach that at the end of this video. Okay, and on the I button too. So that you may watch it previously. Okay, then you should watch this video. Okay, got it. So first of all, what is Carly mutation? See here, the Carly mutant allele, which is denoted by CY, is a phenotypically dominant allele over the wild type wing allele denoted by CY plus. Okay, so though Carly is a mutant allele, but you have to remember that it is a dominant allele, phenotypically dominant allele over the wild type wing allele CY plus. So obviously, in both homozygous and heterozygous condition, the CY phenotypically must show Carly wing phenotype. Okay, so see here in heterozygous condition, CY plus is showing Carly wing phenotype where the wing is curled backwardly. Okay, but interestingly, in CY, these homozygous dominant condition, they are not showing Carly wing. Rather, the progenies are becoming dead, okay, or lethal. Okay, so why this is happening? See here, you have to remember that though Carly mutant allele is phenotypically dominant over wild type wing allele CY plus, but Carly mutant allele is itself a recessive lethal allele. Okay, so that is why in homozygous condition, as the CY is a recessive lethal allele, in the homozygous condition it is showing lethality. Got it? And obviously, as phenotypically CY plus is recessive to CY, so in homozygous recessive condition CY plus will show wild type wing structure in Drosophila. Okay, I hope this part is clear to you now. Now the second thing is, what is 100% female lethality? 100% female lethality means that there will be no female progeny viable in the F1 generation. Okay, so in F1 generation no female progeny should be there. Okay, all female progeny should be killed. So to kill all the female progenies of F1 generation, we must take two lethal alleles L1 and L2 on the X chromosome. If you have seen my 50% female lethality video or 50% male lethality video too, you know that we have taken one lethal allele L. Okay, there. And if you have seen my 100% male lethality video, you must know that we have taken two lethal alleles there, L1 and L2. Similarly, we are taking two lethal alleles L1 and L2 here for 100% female lethality also. Okay. And you have to remember that these L1 and L2 alleles both are recessive to their wild type form, that is L1 plus and L2 plus. Okay. Now, for female lethality, the important thing is that you have to translocate the L1 plus and L2 plus alleles from the father's X chromosome to the father's Y chromosome. Okay, this part is important for female lethality. In case of 50% female lethality too, we have translocated the lethal allele L from father's X chromosome to the father's Y chromosome. Okay, so that 50% females of the F1 progeny can be killed. Okay. Okay, so here we are doing the same thing but with two lethal alleles. Okay, so let's understand this with an example. See here, the problem asks that this is the F1 progeny data and you have to derive the P generation cross from here. So in the F1 progeny data, you can clearly see that no female progeny is there. Only male progenies are there, Carly male and wild male. So you have to generate the P generation cross from here. So obviously no female is there, so the sex ratio will be 45 is to 100 is to 0. That means 100% female are dead here. Okay. Now the phenotypic ratio becomes Carly is to wild equal to 30 is to 15 equal to 2 is to 1. Okay, we know that it is a monohybrid cross ratio for recessive lethal alleles. Okay, and from our previous videos we have learned that in case of monohybrid cross we have to take both the parents' genotype as heterozygous. Okay, so you can see here in the parental generation we are taking mother's genotype as heterozygous. That means one autosome must carry cy, another should carry cy plus, cy plus. Okay, and similarly father is also heterozygous, so cy and cy plus is here. Okay, this is the father. Okay, this is the father. Now the interesting thing remains in the positioning of lethal alleles L1 and L2 over the X chromosome of father and mother. Okay, so see here, if we have taken the X chromosome of father carrying L1 and L2, what will happen? Obviously these two are mutant alleles, so the father will itself not survive, so that could not be possible. So we cannot put L1 and L2 over the father's X chromosome. Okay, so what should we take then? We should take L1 plus and L2 over the father's X chromosome, right? So that father could itself survive. If father does not survive, how will the F1 progenies will be survived? Okay, how will the F1 progenies will be formed? So we must take L1 plus and L2 plus for father on father's X chromosome. Okay, but the problem is here that if we take L1 plus and L2 plus on the father's X chromosome, and if we take L1 plus L2 on mother's 1X chromosome and another X chromosome with L1 L2 plus in complementary condition, what will happen? See here, when these X chromosome will be combined with the father's this X chromosome, it will get the product of L1 plus from both the father's and mother's X chromosome and product of L2 plus from father's X chromosome. So these female progeny will completely survive. Okay, got it. Now similarly, when these X chromosome of the mother will combine with this X chromosome of the father, it will get the product of L2 plus from both father and mother's X chromosome and product of L1 plus from father's X chromosome. So these progeny will also completely survive. Okay, so here you can see both the females are surviving, but in our question there should be no female survived in the F1 generation, so this could not be possible. Okay, so this thing could not be possible. So how can you negate this thing? So to negate this thing, what should you do? You should cut the L1 and L2, L1 plus and L2 plus allele from the X chromosome of the father, and you must translocate this wild type of alleles on the Y chromosome of father. Okay, so that father itself remain viable or remain alive. Okay, because it has L1 plus L2, maybe it is not on the X chromosome but it is on the Y chromosome. Okay, but the F1 progeny females will be dead in this case. Okay, so both the conditions are fulfilled here, so we have to cut the X chromosome. Okay, so that is what I have written here. See here, I have cut the X chromosomes L1 plus L2 alleles from here and translocated it on the Y chromosome. Okay, so that father itself remains viable, as well as when the F1 progeny females will be formed, they could be lethal. Okay, so see here, when this X chromosome of the mother will combine with this X chromosome of the father, it will get only the product of L1 plus, but the product of L2 plus will not be there. So this female will not survive, it will be dead. Okay. Similarly, when this X chromosome of the mother will combine with this cut X chromosome of the father, it will only get the product of L2 plus but not the product of L1. So this female will also not survive, it will be dead. Okay, so that is how you can kill 100% F1 generation females, okay, by translocating the L1 plus L2 plus or the wild type alleles from father's X chromosome to the father's Y chromosome. Okay, so see here why I have written this. Now I think this is clear to you. Okay, why I have written this thing here. Okay, got it. Now let's see what should happen to the male progenies here. Let's take another color from here. Let's take green. So see here. Okay, first of all let me delete all this thing from here. Okay, now see, in case of in case of male progenies, when this X chromosome of the mother will combine with this Y chromosome of the father, it will get the product of L1 plus from both the X and Y chromosomes of mother and fathers, but it will only get the product of L2 plus from father. Okay, because the absence of L2 plus on the mother's X chromosome is complemented by the presence of L2 plus on the father's Y chromosome. So obviously it will get products of both L1 and L2, and this male will survive. Okay. Similarly, this X chromosome of the mother, when combines with this Y chromosome of the father, it will get the product of L2 plus from both father and mother, and the absence of L1 plus in the X chromosome of mother is complemented by the presence of L1 plus on the Y chromosome of the father. So it is getting product of both L1 and L2 plus, L1 plus L2 plus. So this male will also survive. Okay, so that is how males are surviving but females are getting dead in F1 generation. I hope this is clear to you now. Okay, if you have any doubt, you can leave a comment down below. Okay, now another thing may happen that, see, as these L1 and L2 are present on the same X chromosome, okay, so there could be a tendency of recombination. Okay, so if they recombine, what will happen? Obviously recombinant X chromosomes will be formed. So one X chromosome may carry L1, see here, L1 plus and L2 plus. Okay, so one X chromosome will carry L1 plus, L2 plus. Okay, while the other X chromosome recombinant type will carry L1 and L2. Okay, L1 and L2. Okay, so now these two recombinant X chromosome can be formed if recombination is allowed here in the X chromosome of the mother. So now what problem may arise from here? See here, when this X chromosome of the mother will combine with this X chromosome of the father, it will get the product of both L1 and L1 plus and L2 plus. So this female will survive. Okay. And when this X chromosome of the mother will get combined with this X chromosome of the father, it will not get any product of L1 plus or L2 plus. So this female progeny will not survive. But see here, in this case 50% females are surviving. Okay, so but this is not given in our question. No female should survive in the F1 generation. So to stop this, we have to introduce an inhibitor of recombination here on the X chromosome of the mother, so that this recombination process cannot take place in the X chromosome of the mother. Okay, and only then 100% female of the F1 progenies could be dead here. Okay, got it? So now I think why we have taken this type of mutants in the P generation, it is now clear to you. Now let's do the cross. First of all, let's see what gametes could be formed from here. So from here, four types of gametes could be formed. One is CY with L1 plus L2. Okay. Then CY with L1 L2 plus. Okay. Then CY plus with L1 plus L2. Okay. And then CY plus with L1 L2 plus. Okay. So these four types of gametes are formed. And from here also four types of gametes should be formed. CY with this X chromosome, then CY with this Y chromosome. Okay, then CY plus with this X chromosome, and CY plus with this Y chromosome. Okay, so total 8 types of gametes are formed, and when we have put these gametes on the male gametes on the upside and female gametes on the lateral side and done the checker box, we could find the phenotypes of the F1 generation. So see what will be the phenotypes of the F1 generation depending on their genotype. So see here, the first one, carrying CY. So we know that CY is a recessive lethal allele. So whenever there will be CY present, it will be a lethal or dead progeny. Okay, so it is a lethal female. You can see here. Okay, then next it is also carrying CY, so it is a lethal female. Okay, then this is carrying CY plus, so it should be a Carly female as it is carrying 2X chromosomes also. But you can see here, though here is the presence of L1 plus, but L2 plus is absent. Okay, so due to absence of L2 plus, it is lethal female. Okay, got it. Next, similarly here, that due to absence of L1 plus, it is a lethal female. Okay. Next, this is a lethal male due to presence of CY here. Then this is also a lethal male due to presence of CY here. It could be a Carly male, and it is a Carly male. Why? Because you can see here that the male carries L1 plus alleles in both X and Y chromosome, and L2 plus allele is in its Y chromosome. So the product of both L1 plus L2 plus is here. So it is a viable Carly male. Similarly, it is also a viable Carly male because it has both L1 plus and L2 plus alleles and their products. Okay. Now see, it could be a Carly female, but it is dead due to absence of L2 plus allele. So it is a lethal female. Okay. Next, it could be a Carly female, but it is dead due to absence of L1 plus allele. So it is a lethal female. Then this could be a wild female, but due to absence of L2 plus, it is a lethal female. It also could be a wild type female, but due to absence of L1 plus, it is a lethal female. Okay. Now see, this is a Carly male due to presence of both L1 and L2 plus alleles. Okay, so it is a Carly female male, and similarly it is also a Carly male due to presence of both L1 plus and L2 plus alleles. Okay. Now see here, as it is carrying CY plus, so it is wild type, and as it has X and Y chromosome with both L1 plus and L2 plus allele, so it is a surviving wild male, and similarly it is also a surviving wild male. Okay, so from here you can see only how many progenies are surviving. A Carly male, second Carly male, third Carly male, fourth Carly male, 4 Carly males and 2 wild males are surviving. So among 16 progenies, only 6 are survived. Okay, and among these 6, 4 are Carly male and 2 are wild male. Okay, so in the what are the 4 Carly males? In box 7, box 8, box 13 and box 14 Carly males are there, and in box 15 and 16 wild males are there. In our given question, the sex ratio of Carly male and female was 30 is Carly male and wild male was 30 is to 15, that is 2 is to 1. Okay, so 2 is to 1, and let's see this ratio is similar in our observed checker box or not. So it was given Carly male is to wild male equals to 30 is to 15 equal to 2 is to 1, and here we can observe that 4 Carly males are there and 2 wild males are there, so it is also 2 is to 1. So our cross is correct, it is now proved. Okay, because given and observed values ratios are same. Now let's take a look on the calculation of 10 lethal progenies of F1 generation, as 6 are surviving, so obviously 10 will be lethal. Among these 10 you can see 2 are lethal males. Okay, so see where are the 2 lethal males? See box 5 and box 6 are showing lethal males. Okay, lethal males, and they are lethal due to presence of CY. Okay, so due to CY or homozygous recessive lethal allele Carly, they are lethal. Okay, now obviously 8 will be lethal females. Okay, now among these 8 lethal females, there are 2 lethals which are lethal due to presence of CY. Okay, that means recessive lethal allele Carly. They are present in box number 1 and 2. Let's see where they are present. See here in the box number 1 and 2, due to presence of CY in both the cases, these females are lethal. Okay, now see, due to absence of L1 plus, 3 females are dead. Okay, and they are present in box number 4, 10 and 12. Okay, 4, 10 and 12. Let's see in the box number 4, this female is lethal due to absence of L1 plus. Okay, and if it survived, it could be a curly female. In box number 10, see here, this female is dead due to absence of L1 plus. So it is also a lethal female due to absence of L1 plus, and if it survived, it could also be a curly female. Okay, and see here in the box number 12, this female is also dead due to absence of L1 plus, but if it survived, it could be a wild female. So these 3 progenies are dead due to absence of L1 plus. Among them, the box number 4 and 10 shows that if they survived, they could be curly, and box number 12 shows that if it survived, it could be a wild female. Now, due to absence of L2 plus, there are 3 females which are being dead, and they are present in box number 3, 9 and 11. Okay, so see here in the box number 3, you can see that due to absence of L2 plus, this female is dead, and if it survived, it could be a curly female. Okay. Now in the box number 9, let's see, this female is also dead due to absence of L2 plus, and if it survived, it could be a curly female. And in box number 11, see, this is also dead due to absence of L2 plus, but if it survived, it could be a wild female. Okay, so in the box number 3, 9 and 11, we can see these progenies are dead due to absence of L2 plus, but if they survived, the progenies of box number 3 and 9 could be a curly female, and the progeny of box number 11 could be a wild female. Okay, so that is how total 8 females are dead here. Okay, all the females are dead here. Among 16, 8 were females, and all of them are dead. Okay, due to either absence of L1 plus or L2 plus, or due to CY. Okay, got it? So this is all about the 100% female lethality genetic cross in case of curly mutation in Drosophila. I hope that this video is clear to you. If you have any doubt, don't forget to leave a comment below. And if you like this video, don't forget to hit the like button and share it with your friends. And don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.